This is the special senses, which is chapter 15. There are 5 special senses: vision, taste, smell, hearing, and equilibrium. First one we're going to talk about is vision. Vision is the dominant sense. And yes, it's true that if something happens to one of the senses, that the other ones will become much more sensitive and more acute to kind of take over. So if you become blind, your hearing becomes much better. And so they work together, obviously. The eye has 3 different layers. It has the lens, it has humors, which is the fluid that's inside of it, as well as accessory structures. If we look at the accessory structures, excuse me, the eyebrows are there to help protect the eyes from sunlight. The eyelids are for protection. The conjunctiva helps prevent the eye from drying out. The lacrimal apparatus, we'll look at that in just a moment, but it helps to protect the eye by lubricating it, moistening it. It involves your tears, which contain a chemical in it that helps to protect from infection. And then you've got your extrinsic eye muscles, which are involved with controlling the movement of the eye itself. So here's the lacrimal apparatus. The lacrimal apparatus, like I said, is going to be producing the tears. Now, a lot of people think the tears are produced here in the inner corner. This is the right eye, so the nose is right here. So a lot of people think, well, that's where I see the tears. That must be where it's produced. No, it's not. You have a lacrimal gland up here in the upper outer corner of the eye that actually produces the tears. And it's going to secrete them. Every time you blink, it's going to flush those tears across the eye, and it's going to go from the outer upper corner across the eye to the lower inner corner of the eye. Like I said, that helps to lubricate the eye, help moisten it. Now, right here is the pupil of the eye, the hole in the middle that allows light to come in to the eyeball itself and hit the retina in the back to stimulate it so you can see. That is an opening, and if you get in the eye, then you've got the nerve leaving the eye in the back, the optic nerve, and that potentially could be a route into the brain, which you don't want any foreign substance like bacteria to get in that way. So the tears contain a chemical lysozyme that is bactericidal, which means it will kill bacteria. So it's a protective mechanism as well as moistening and lubricating. So every time you blink, it's flushing over the eye. It's going to drain right here in what's known as the lacrimal punctum. So it's going to flow down in the lacrimal canal into this area of the lacrimal sac, which can help hold the tears, and then it's going to flush down the nasolacrimal duct and drain into the nasal cavity. So if you're saying, well, how come I always see my tears here? Well, normally every time you blink, you're not crying all the time. It's able to drain. But if something happens, some emotional trigger, or you're cutting onions, or whatever the case may be, where you start generating a lot of tears, more than what can quickly drain down through the lacrimal canal. This is a small opening. So if you're producing more than what can drain down here, it backs up, and it's going to flow over and down your cheek, and you're going to be crying. The lacrimal sac, notice right here, if you were to get a blow to the nose, it's going to push against this, and so some of the tears that are stored here, because it doesn't always empty completely, it's going to flow down here. You're going to get a runny nose, and some of it's going to flow back, basically, and be squished, pushed, squeezed out here. So when you get a blow to the nose, yes, you're going to get a runny nose, and you're going to be crying. Why do you get a runny nose every time you cry? Because it's draining down into the nasal cavity. Your extrinsic eye muscles, there are 6 of them. They help to move the eye left, right, up and down, and kind of at angles. And so the extrinsic means it's on the outside of the eyeball. They're helping to secure, hold the eyeball in place, and allow for that movement. So now if we look at the structure of the eye itself, as I said, there's 3 layers. There's the fibrous layer, vascular layer, and the inner layer, which is the retina. You have the humors, which are the fluid, the liquid inside the eye. There's the aqueous humor and the vitreous humor, and then the lens. So when we look at the eye here, excuse me, the front right here, you've got the cornea. The light's going to be coming from this direction, pass through the cornea. You have a front chamber, as we'll talk about in a moment, the posterior chamber. The light passes through the pupil. The pupil is that opening there to allow the light to come in. This is the lens. The light will pass through the lens. It's now going to pass through the back portion of the eye, which is the posterior chamber, through this fluid, which is the vitreous fluid, and it's going to come back here. And this yellow layer is the retina. That's where your photoreceptors are. That will be stimulated. And then the axons will all congregate here. This is the optic nerve. This yellow here is the optic nerve. Notice you do have a good blood supply here, so you have a central artery and vein that are coming in, supplying the blood nutrients. The optic disc is this area right here where these axons all congregate and leave as the optic nerve. There's no photoreceptors right here, so that's often commonly referred to as the blind spot. Directly back from where the pupil is, the center, that is known as your fovea centralis. It's just known, it's the center of the back of the eye. We look at the three layers. The fibrous layer, this is the outermost layer. Excuse me. The sclera is the posterior portion. That's the white of the eye. The cornea is a very small portion in front. It is transparent to allow light to pass through so light can enter the eye. The cornea does have nerve endings, but it has no blood supply. Because there's no blood supply, you can have a cornea transplant and you don't have to worry about any rejection issues with it. One thing with the cornea, you do need to be careful because there are those nerve endings you often feel. Like if you get an eyelash on your eye, you can feel it. You want to be careful if you get something in your eye not to scratch the cornea. So you don't want to do that. The middle layer is the vascular layer. It is pigmented. There's three different regions of it: the choroid, the ciliary body, and the iris. The choroid is in the posterior part of the eye. It has a very rich blood supply. It's pigmented to help absorb light. It does have the opening for the optic nerve to leave the eye. The ciliary body surrounds the lens. It helps to hold the lens in place, and it helps to control the shape, because the lens in a nice healthy eye, the lens is going to change shape depending on what you are looking at and how far away that object is, because it has to change its shape to focus the image on the back of the eye. So you have to have something to hold it in place. The iris is the colored part of the eye that you see. It's actually smooth muscle, so you have no control over whether it contracts or not. When it's constricted, this occurs in very bright light, so it decreases the amount of light coming in. It's usually also with very close vision. The pupil will dilate in dim light to try to increase the amount of light coming in. So the pupil is the center, that hole in the center that allows the light in. So the iris is going to be contracting and relaxing, changing the diameter of that pupil. The color is determined genetically. And just so you know, they do not put the eye color on the birth certificate for a newborn, because within the first 6 months after birth, the eye color may change a little bit. Usually by 6 months, they'll say, okay, whatever color the eyes are, that's it. That's what they're going to be. But it can change in those first 6 months, so they won't put it on the birth certificate. The retina is the innermost layer. This is what contains your photoreceptors. It contains neurons and those supporting glial cells. There are two layers. The outer layer of it is pigmented, and the inner layer is the neural layer. So that pigmented layer is only one cell layer thick, very, very thin, obviously. It helps to absorb light. It prevents it from scattering all over the place in the eye. It also does contain phagocytes. Phagocytes are those cells that They're cleanup crew. They can recognize anything that is a foreign substance and basically chew it up. I used to say it's kind of like Pac-Man, but most of you now probably have no idea what Pac-Man is. So it's going to eat anything foreign. And once again, it's kind of a protective mechanism because the pupil is an opening. You don't want to take a chance of having, say, bacteria enter the eye through the pupil. And the idea in terms of defense with the body is, number one, prevent anything from entering in the first place. So you have the tears. But if something enters, let's contain it and not allow it to penetrate even deeper. So the phagocytes are there so that if something did enter the eye, let's destroy it and not allow it to enter the brain by moving through the optic nerve. This pigmented layer also contains vitamin A. The neural layer is composed of the photoreceptors, bipolar cells, and ganglion cells. As I mentioned earlier, the optic disc, that's where all the optic nerve leaves the eye. There are no photoreceptors there, so that's the blind spot. So what's going to happen is the light's going to come in. It's going to activate the photoreceptors, which in turn stimulate and activate the bipolar cells, which in turn are going to stimulate and activate the ganglion cells. And then all those ganglion cells, all their axons, are all going to come and congregate and leave the eye together in a group as the optic nerve. So if you are looking at the eye, if this is the eye here, right here would be the interior part of the eye. The light's coming in through the pupil, enters in the back of the eye, and so it's going to come in this area. Boom, here's your retina right here, which is enlarged up here. So the light comes in. Here are your ganglion cells, these gray ones. These middle pink ones are your bipolar cells. And then back here we have our photoreceptors, which are the rods and the cones. That's the neural layer. Here, this dark blue back here is the pigmented layer of the retina. So the light, it's going to come and just bypass the ganglion and bipolar cells. Doesn't matter about this. Come back here. Remember, the pigment helps keep the light from scattering. It's going to activate, the light activates these photoreceptors here. It activates both the cones and the rods. They in turn activate the bipolar cells, which in turn now activate these ganglion cells. And the axons, see each one of these, you've got your axon, they're all going to start coming, and that's what is yellow in this diagram, are all those axons from the ganglion cells, and they all come together and congregate back here and leave together in this big group, which is the optic nerve. And right here at the optic disc, notice you don't have any of these photoreceptors right in here because that's where all the axons are leaving. So this is the optic disc or the blind spot. The photoreceptors, you have rods and you have the cones. Rods allow you to see in dim light and they allow you to see black and white. The cones are better stimulated in bright light and they allow you to see in color. There are internal chambers that are filled with fluid in the eye. Pretty much the lens and the ciliary zone, you'll divide that eye into two chambers. The posterior chamber is filled with vitreous humor, which is a gel-like, kind of jelly type substance. That is formed during embryonic development. So what you have is what you have. You can't replace that. The anterior chamber is filled with aqueous humor, and that is formed and drains continuously. So that is constantly being replaced. One thing in any eye exam they will do is they will monitor the pressure of these fluids. So you don't want to get too high. It needs to be within a certain range. If you have increased pressure in the eye, that is glaucoma. So they want to monitor that. So in this picture back here is the posterior chamber. You have the vitreous humor there. It really is kind of like a Jello type consistency. Here's your lens right here. Here's your iris. The cornea is up here. So the aqueous humor is going to be produced back here. It's going to move then in front of the lens, through the pupil, and down front, and then drain here through the sclerovenous sinus. So it's constantly flowing and being replaced. The lens itself is biconvex. It's transparent. It has to be transparent in order for the light to pass through. If it starts to get cloudy or opaque, that is cataracts. It's avascular. So once again, you can have surgery and replace the lens without fear of rejection. Like I say, that would be cataracts. It's flexible. It has to change its shape so it can project that image back on the retina so you can see it. And it's held in place by the ciliary zonule. So this is showing how the lens is going to be changing its shape depending on where you're looking, so that no matter whether the object is further away or close up, you are still projecting that image on the back of the retina. So notice how the lens is going to change its shape. For distance, usually it's flattened out and then it tends to bulge, kind of short and squatty, when you're looking at something close up. The other thing I'm just going to point out right here, notice that it's part of physics. The light waves, when it goes through any type of a lens, it's going to flip it upside down. So the image that is projected on the back of the eye is actually upside down. That upside down image is going to be sent to the brain. Your brain knows automatically to flip it back right side up. Emmetropia is normal vision. Myopia is nearsightedness, so the object's being focused in front of the retina. Hyperopia is farsightedness, so the object is focused behind the retina. So myopia and hyperopia is going to be blurry. Astigmatism is where you have an unequal curvature of either the cornea or the lens, and this can be accommodated or corrected with the lens, how they do the shape of your lens if you wear glasses or contacts. And then accommodation is the ability to focus on really close objects. So this is showing with emmetropic eye, which is normal vision. The myopic eye, it's focusing too close, and then this is too far and back. So how do you correct these? You correct it with your lenses. The shape of the lenses is going to help direct so that image is now focused on the back of the eye, on the retina where it needs to be. Your photoreceptors are modified neurons. The rods, as I said earlier, are for black and white. They're very sensitive. Cones need bright light for activation. This allows you to see in color. There's 3 different types: blue, red, and green for the pigment. Now, if you're colorblind, that is a genetic issue. There's a gene that allows you to see these different pigments, or not see them if you're colorblind. Phototransduction refers to, okay, how do you get this light energy, the light that's coming in the eye, how do you get that converted to an action potential so it can be sent to the brain? The light energy has to be converted first to a greater potential. Vitamin A is going to help play a role with this. It is going to help form the compound retinol, which then is going to combine with opsin to form this compound rhodopsin. The rhodopsin is actually going to absorb the light, and it's going to start the whole process of generating a greater potential. So vitamin A is involved. Where do you get vitamin A from? Naturally, vitamin A is often found in your yellowish orange vegetables like carrots and sweet potatoes or yams, things like that, squash. So your mom was right when she said eat those carrots to help your eyesight. There is some truth actually to that. The like I say, the rhodopsin is going to help generate this greater potential in the photoreceptors, which are then going to generate a stimulate the bipolar cells, which then generate a greater potential. And then they're going to stimulate the ganglion cells, which have the action potential that's then going to be sent to the appropriate visual cortex area of the cerebrum. So this is just showing, here comes the light. It's going to help to activate the opsin with assistance from vitamin A, and you have this stepwise process so that you get this greater potential generated. Now, once the ganglion axons, those all, remember, congregate, they leave the eye, forming that optic nerve. As the optic nerve travels, it's going to pass through the optic chiasma. Now you have all those axons there. Now, the medial nerves from each eye will cross over to the opposite side of the brain. The lateral ones stay on the same side, but the medial ones will cross over in the optic chiasma. Then they continue to the optic tracts. The optic tract is going to move around the hypothalamus to the thalamus to the visual cortex. For depth perception, you have to have the visual field from the two eyes overlap. Now, they overlap significantly already, but they're at slightly different distances that you're looking at. And in the visual cortex, what happens there, it's going to fuse the two images from both eyes together, and that then allows you to see. Now, you might think you're looking at the same distance, but you're actually a little off from one to the other eye, and that allows for the depth perception. So if you see on this particular diagram here, we're looking from the bottom side of the eye. So this is the right eye, or the brain, this is the right eye, and then the left eye. So the blue area and green area is what you see from the right eye, and then the yellow and green area is what you see with the left eye. So that green obviously is the overlap area. What happens in terms of the pathway that the nerves take? Here's your optic nerves leaving. So from the right eye, they're blue, yellow from the left eye. They come here. Right here is the optic chiasm. This is where I was talking about. The medial nerves are going to cross over to the other side of the brain. So obviously we're in the cerebrum right now. It's going to cross over. The lateral nerves stay on the same side. So this is going to cross over and then continue on through here to the visual cortex area. So this is the crossing over in the optic chiasma that we're talking about, and then this is the optic tract afterwards. The brain in the visual cortex knows to fuse these two images together so that you have that depth perception. Now, just so you know, some people don't have depth perception. The way my eyes work, I can see out of both eyes, but I only see out of one eye at any particular time, and I just constantly switch. So I'm either seeing this or I'm seeing this, and I just switch back and forth. It's subconscious as far as the switching. I'm usually not even aware of it. If I want to, I can control which eye I'm looking at, but usually I don't. Because I'm only seeing out of one eye at a time and I'm constantly switching, I do not have these two images to overlap. Therefore, I do not have depth perception. Why does this happen? Who knows? I mean, I was born with some eye issues, whether it's from that, who knows? But for me, I don't consider it a disability because for me, this is my normal. This is all I've ever known. How do I judge depth perception? For some things, yeah, it's a little tough for me to see what's in front there. Some things, just because this is all I've ever known, I've just kind of adapted and learned to kind of look at shadows and how things look relative to each other. I just kind of subconsciously am able to figure out, but there are times when no, I have no clue. Items, say which ones are closer, which ones are farther away. I will say this, most things look much closer than they actually are. So don't panic if you ever see me driving and I'm, say, pulling up behind you. I will not hit you. Things look much closer. So it drives my sister nuts when she rides with me because she lived for many years in California and is used to all the traffic there. Well, I tend to leave probably at least 8 feet between me and the car in front of me. So don't worry, I won't hit you. But it's just kind of it is what it is. The chemical senses, these are the senses of smell, which is olfaction, and taste, which is gustation. They involve chemoreceptors, which are going to be responding to chemicals. They must be in an aqueous solution, however. Olfaction, once again, this is the sense of smell. The location of the receptors is going to be in the roof of the nasal cavity. When you sniff, that's drawing more air into the nasal cavity. It allows it to swirl around more, which increases the chances of activating those receptors. The receptors are ciliated bipolar neurons. The chemical must be in a gaseous state when it enters that nasal cavity, and then it gets dissolved to activate the receptor. It gets dissolved in mucus. The olfactory neurons do have a range of sensitivity to a variety of different chemicals. What's going to happen is, excuse me, as you breathe and sniff in air, it's going to swirl around through the nasal concha here, swirling around. The receptors are located up here in the top or the superior portion of the nasal cavity. And notice that you're producing some mucus along here. You have the ciliated cells. So as you can see right here, the gas is going to, as it comes in, it becomes dissolved in the mucus, and it's going to activate those receptor cells here, which then come up here, and they will then activate the cells in the olfactory bulb, which is right here, and then send it down the olfactory tract. So the olfactory receptors are activated, sends olfactory bulb to the mitral cells, the olfactory tract, which then sends to the olfactory cortex area of the in the cerebrum is where we're talking about. Now the nerves are going to pass through the limbic system, so that's going to trigger an emotional connotation with that smell, good, bad, or indifferent. An example that I often tell is my mother, who was a wonderful, excellent cook. For some reason, whenever she made Boston baked beans, that was the one thing she would just burn the daylights out of it. And if you've ever smelled burnt beans, oh, it's not good. And it is not good to have to clean that pan. So as a kid, my sisters and I would always have to wash the dishes, after dinner. And so you always are hoping it was not your turn to wash when she made those, because it was bad. I still associate, when I hear just even Boston baked beans, I can just smell that burnt smell, and it's just kind of this dread of, oh my gosh, it's going to be awful cleaning that pan. It's a nightmare. So I just have this emotional connotation with it. Several years ago, I was in Boston for a conference, and Boston, all these restaurants would be advertising their Boston clam chowder and then Boston baked beans, and the chowder, I will say, was great. The Boston baked beans, I couldn't order it. I couldn't eat it. I just have this, we'll say I have an emotional connotation to it. It's there in the memories. Once again, this is just showing how the chemical has to be dissolved, in this case in the mucus, to bind to the receptor, and then you get this cascading event where you activate the cyclic AMP, and then ultimately triggering depolarization. Gustation, this is the sense of taste. It involves the taste buds, which are the sensory organ. Most of those are located on the tongue. Once again, the food has to be dissolved in order to stimulate those receptors. There are 5 taste sensations: sweet, sour, salty, bitter, and umami. The nerves are also going to pass through the limbic system for gustation, so you're also going to put in emotional context with taste. It has been estimated that 80% of taste is actually smell. The two are very closely related. You all have noticed this whenever you have a cold or you're congested. If you ever notice, you think, oh, the food doesn't taste as good or it has no taste at all. You're still cooking the food the same way. It's just when you can't smell it because you're all congested, so you can't activate those receptors in the nasal cavity, so you can't smell. Yes, it has an effect on what you perceive the taste to be. It's going to be very bland. The other thing that has a huge role with taste is vision. This is an example, really shows how the special senses work together. When you see something, just by the look of it, you automatically start to form an idea of what it is going to taste like. Vision, as we said earlier, is one of your dominant senses. It's going to play a huge role. So if it looks pretty, the presentation of it, that's why, you know, at fancy restaurants, they learn that you just make it look pretty, you're automatically going to think, oh, this must taste good. Look how pretty it is on the plate. For some people, that's important. In terms of vision, this is something that you can do. I've done this for years in face-to-face labs. And you can easily do this at home. If you take flavored water, which if you get the kind that's clear, remove the tag and label the bottles like A, B, C, whatever. Now you can have your little cheat sheet on the side so you know what it is. And then you put food coloring in it. If you see, let's say, you see a drink that is colored red, you're probably automatically, without smelling it, without tasting it, you just see it's red, you're automatically going to assume, oh, I bet that's maybe strawberry or cherry. Some people may think watermelon flavored, maybe red apple flavored. If it's red, you're not going to guess, oh, it's orange flavored. You're not going to guess, oh, I bet that tastes like lemon. It's the wrong color for that. So as soon as you see something, color is a huge thing. As soon as you see it as a certain color, your brain automatically says, oh, I bet this tastes like something that's red. So if you take flavored water, like I used to do, and let's say you take a lemon flavored water and you put blue food coloring in it. Take lemon flavored water, so you put blue in it. You take orange flavored water and make it green. Strawberry flavored, make it yellow. Change the colors up. And then have your friends or kids or whomever, have them taste test it. See if they can guess what the flavor is. And sometimes, some people are pretty good at guessing. Sometimes it might take a while to guess it. My kids have now learned that if I'm teaching face-to-face, they kind of learn when I get to this section, and because I tend to bring the leftovers home, and they're like, oh, they don't trust now with flavored water. They know, oh, if it's colored, it's probably not what I'm expecting. The other thing that you can do, and I've done this as well, take lemon flavored Jello that you make yourself, the powdered. And so when you're mixing it up, you know, just make the real easy where you just mix it with a, what is it, a cup of hot water. And you add, if you get lemon flavored, add red food coloring to turn it orange. Let it set, and then have people taste that. And they see it's orange, and automatically they're going to go, oh, this is orange. And you start to eat it, and initially, yeah, it's orange. And then this, after you're like, wait a minute, this has a little bit different flavor. Maybe it's tangerine. You are not expecting something that is lemon flavored to be orange. And it's a really powerful thing to show how strong the vision is, in that it basically it's messing with your brain trying to override. This is not what I'm tasting, per se, but it looks like, because of the color, it should be this other flavor. And then if they can smell it, then that's going to be another thing that's going to kind of throw things off. So 80% of taste is smell. So this is showing on the tongue of your taste buds. This is why the taste buds are down here. So they're down lower in these little openings. That's why you have to have it dissolved, be able to reach down here in order to activate the taste buds. That's another thing you can do, is compare a solid versus something liquid. If you take a sugar cube and put it on the tongue, you're not going to taste the sweetness until that sugar dissolves in your saliva. That's part of what your saliva is doing, is dissolving the food so you can taste it. So if you put a sugar cube, it's going to take a little bit longer for you to taste that sweetness versus, say, you drink a soda or a sweet tea, which is sugar is already dissolved. You're going to taste that right away. So here the sweetness is usually in this front portion, salty in here, sour. In the back is bitter. One of the reasons why your taste buds for detecting bitter is in the back here is because a lot of your natural poisons have a natural bitter taste to them. And having your bitter taste buds back here, this often triggers the gag reflex where you want to spit it out. That's kind of a survival mechanism for you, a protection mechanism, so you don't swallow the poison. So you'll find that some things that naturally are okay to eat, that naturally have a bitter taste to them, oftentimes sweeteners are added to kind of counteract that natural gag reflex. Hearing, excuse me. The ear is divided into the external ear, the middle ear, and the inner ear. All three parts of these are involved with hearing, and then the inner ear is also involved with the special sense of equilibrium. The external ear is composed of the auricle or pinna. This is going to help funnel sound waves into the next part, which is your external acoustus medius, which is also known as your auditory canal. So the auricle, which is the part that you see, is going to funnel those sound waves into that auditory canal, and it extends to the tympanic membrane, which is commonly referred to as the eardrum. That is what divides the external ear from the middle ear, and when the sound waves hit that, that's going to start vibrating. So here is the external ear. As I said, it's going to extend from the auricle all the way to the tympanic membrane, which is right here, kind of this turquoise colored structure right there in this diagram. This area right in here is the middle ear, and then these structures over here are comprising the inner ear. So inner ear, middle ear, and external ear. If we look at this enlarged picture right here, you've got the external auditory canal with the tympanic membrane right there. So when the sound waves hit that, that will start vibrating. When that vibrates, it's going to start to move and vibrate these three bones, which are known collectively as the auditory ossicles. Those chambers should be filled with air. As they vibrate down here, they're going to start a vibration here to send here to the cochlea, which the vibrations are going to travel in and then back around and then hit the round window. So we're going to look at all these a little bit more detail. So that middle ear should be filled with air. If you have an ear infection, the middle ear infection, usually if you have an ear infection, it's with the middle ear. Usually what happens is you have gotten fluid in there, it's nice and warm, there's fluid there, and it makes a wonderful environment for bacteria, for viruses to be in there. And so when you have an ear infection, it's actually filled with fluid. As I said, the tympanic membrane will vibrate, and it's transmitting those vibrations to those auditory ossicles, which are going to amplify those sound waves now. The three auditory ossicles, they are the malleus, the incus, and the stapes. And so here when we're looking we've got the tympanic membrane. And so that gets vibrating, the malleus, the incus, and the stapes right here. And as this vibrates, it's going to, you can see here the muscles that can move as those move. Here is your pharyngeotympanic tube, also known as your Gustation tube. And this basically is going to connect down to the pharynx. When you swallow, you often hear the popping noise here as you're trying to equalize the air pressure in here. The internal ear, and here you have the bony labyrinth, which contains perilymph. It is composed of the vestibule, which is subdivided into the saccule and the utricle. You also have the semicircular canals and the cochlea. The cochlea contains the cochlear duct. It looks like a shell of a snail. It contains the organ of Corti. That is where the hearing receptors are going to be located. And then you also have a membranous labyrinth, which contains endolymph. Excuse me. So here is the end of the stapes. It's connected to the oval window. So the external ear is funneling the sound waves in towards the middle ear. The tympanic membrane gets vibrating. The middle ear is going to amplify those sound waves. And then in the inner ear is where the receptors are to actually convert that then into the sound waves into generating an action potential, so that then you can send that message down through the nerves to the auditory cortex to interpret the sound. The inner ear, the cochlea right here, the one that looks like the snail shell, this is where the receptors in here, in the spiral portion of it, is the organ of Corti. That's where the receptors for hearing are located. The receptors for equilibrium are going to be involved out here in the vestibule area. You have the saccule and you have the utricle, and it's going to involve the semicircular canals, which are over here, are involved not with hearing but with equilibrium. So this is showing with the cochlea what's going to happen. Excuse me, is that you've got the oval window. Now this is showing it kind of stretched out. You do have these different vesicles where the sound waves are going to be moving. It gets the length moving, the vibrations going. Right here in the center portion is going to be the organ of Corti. So the sound waves, they funnel through that external acoustic media, triggering the tympanic membrane to vibrate. So then those three auditory ossicles, those three bones, they amplify the vibrations. The stapes, which is connected to the oval window, it triggers the oval window to start vibrating. As the oval window vibrates, you get these pressure waves that are going to move the fluid that's in those open canals of the cochlea that we just saw in the previous picture that I pointed out, the scale of vestibuli. It's going to get the fluid moving through there. So it's going to move around that cochlea, move around through the snail shell, if you will, to the innermost part, and then it's going to move back. And as it moves, it's going to stimulate the hairs that are located in the organ of Corti. As it stimulates those hairs to move, then that is going to generate an action potential. So this is showing the sound waves. The outer ear, here in the middle ear, they become amplified, and then in the inner ear, now you get it moving through the fluids of the cochlear canals, stimulating the organ of Corti. So each auditory cortex is receiving impulses from both ears. The tone is going to be perceived separately. You're able to detect the pitch, the intensity, and the location of where it's coming from. Equilibrium. The vestibular apparatus, the equilibrium receptors are going to be located in the semicircular canals and in the vestibule of that inner ear. The vestibular receptors, they can monitor both the linear acceleration, how fast are you moving, and also what is the head position. Is it upright? Is it sideways? The semicircular canal receptors, they can monitor changes in the rotation of the head. So tilting back, is it tilting forward, etc. For being aware of your balance and your orientation, which is going to involve equilibrium, it involves receiving information from these receptors in the vestibule, the inner ear, but it's also going to involve getting information from receptors from visual. What are you seeing? Are things upright? Are they sideways? Whatever. And then the somatic receptors from these are going to be, there are receptors in like your skeletal muscles and your joints, and all these play a role together to help know what is your orientation.